Hello everyone. Today we will discuss about DNA replication in prokaryotes. Replication is the process of producing two identical replicas of DNA from one original DNA molecule. It is the process of producing two identical replicas of DNA from one original DNA molecule. Let's see some of the features of replication or characteristics of replication. First feature is this replication is semi-conservative. Semi-conservative. Semi-conservative means when a new double-stranded DNA molecule is formed, one strand will be from the original template molecule and the other strand will be newly synthesized. So, when a new double-stranded DNA molecule is formed, in its one strand, it is from the original template strand or original template molecule and the other strand will be newly synthesized. Or we can say each strand in the double helix act as a template for the synthesis of a new complementary strand. This is semi-conservative method. This theory was confirmed by Meselson and Stahl experiment. So, in Meselson and Stahl experiment, bacteria were grown in a medium that contained N15, that means heavy isotope of nitrogen, when all the DNA were labeled with nitrogen. So, here it is. It is N15 labeled, that is heavy isotope of nitrogen. Then these cells were allowed to divide in a medium containing N14. Then these were allowed to grow in a medium containing or divide in a medium containing N14. This is N14. So, in the first generation, all DNA molecules were half labeled. That means both N15 and N14. This is the first generation. So, it will be half labeled N15, N14. This is the first generation. Then in the second generation, equal number of half labeled and completely labeled molecules were present. Equal number of half labeled and completely labeled. So, it will be like, this is the first generation. In the second generation, it will be like this is half labeled, that is N15, N14. Then it is N14, N14 and N15, N14. So, this is Meselson and Stahl experiment. So, bacteria were grown in a medium that contained a heavy isotope of nitrogen, that is N15. Then these cells were allowed to divide in a medium containing N14. In the first generation, all DNA molecules were half labeled N15, N14. In the second generation, equal number of half labeled and completely labeled molecules were present. So, that is the first feature of replication, that is semi-conservative. Second feature is replication is bidirectional. Replication is bidirectional. Bidirectional means replication is in two directions at the same time. It is in two directions at the same time and it will be in 5' 3' direction. That is bidirectional. Third one is it is a semi-discontinuous. Semi-discontinuous. That means replication occurs continuously on one strand and discontinuously on the other strand. In one strand it is continuous and in the second strand it is discontinuous. That means that is called semi-discontinuous. Then fourth feature is high fidelity. Fidelity means the ability of the enzyme Dna Polymerase to correct errors in the newly synthesized DNA strand. That is known as fidelity. It means the ability of the enzyme Dna Polymerase to correct errors in the newly synthesized DNA strand. The next feature is DNA replication is extremely accurate. Then replication is very rapid. That is about 750 to 1000 base pairs per second. So it is very rapid. Then DNA replication begins with the unwinding of two anti-parallel complementary strands. So it begins with unwinding of two anti-parallel complementary strands and it results in the formation of two single strands. So these are the salient features of replication, that is semi-conservative, bidirectional, semi-discontinuous, high fidelity, then accurate, very rapid, then unwinding of two anti-parallel complementary strands and it results in the formation of two single strands. These are the salient features. So in the process of replication, DNA polymerase enzyme, this enzyme adds nucleotides one by one to the growing DNA molecule that is complementary to the template strand. So the addition of nucleotides requires energy. This energy is from ATP, GTP, TTP and also from CTP. In prokaryotes, there are three main types of DNA polymerases. DNA polymerase 1, 2 and 3. In this DNA polymerase 3 is the enzyme required for DNA synthesis. 3 has 3' 5' exonuclease activity. 3' 5' exonuclease activity, it is required for DNA synthesis and it has highest polymerization rate. In the case of DNA polymerase 1, it is required for the removal of RNA primer. It has 3' 5' and 5' 3' exonuclease activity. 3' 5' and 5' 3' exonuclease activity. Its main function is gap filling and synthesis of lagging strand. This is DNA polymerase 1. In the case of DNA polymerase 2, it has 3' 5' exonuclease activity. This DNA polymerase 1 and 2, these are required for repair. So this DNA replication involves the action of various specialized enzymes and proteins. First enzyme is DNA polymerase. Second enzyme is primase. It is also known as DNAG. So this primase is DNA dependent RNA polymerase. DNA dependent RNA polymerase. Its other name is DNAG. It synthesizes RNA primer. That is the function of primase. It synthesizes RNA primer. Next enzyme is helicase or DNAB. This helicase causes the unwinding of DNA helix, or it opens the DNA helix. This is by breaking hydrogen bonds. By breaking hydrogen bonds, it unwinds the DNA helix. That is helicase or DNA B. Next one is topoisomerases. Topoisomerases. Two types are there, 1 and 2. 2 is known as DNA gyrase. This topoisomerase 2, this relieves the torsional strain. It relieves torsional strain that results from this helicase induced unwinding. When there is unwinding by the helicase, there will be torsional strain. This is relieved by topoisomerase 2, that is gyrase. The next enzyme is ligase. This ligase enzyme seals the gap or nick, gap between the Okazaki fragments. So these are the main enzymes. Then proteins are, first one, DNA A protein. It is the initiator protein. Then single stranded binding protein. Next one is single stranded binding protein or SSB protein. This single stranded binding protein binds with the single stranded regions of DNA in the replication fork. It binds with single stranded regions of DNA in the replication fork, and it prevents the strands from rejoining. So these are the main enzymes and proteins in the replication. So let's see the steps in replication. Mainly 3 steps. That is initiation, elongation and termination. Initiation, elongation and termination. So first we will see initiation. Replication of DNA. DNA replication begins at a single point, that is known as origin of replication. Origin of replication or Ori C. So DNA replication begins at a single point known as origin of replication or Ori C. This is AT rich. In E. coli, the Ori C consists of 245 base pairs. It consists of 245 base pairs of DNA. This 245 base pairs consists of 2 short sequences. It consists of 2 short sequences. That is, one is 4 repeats of 9 base pair and second one is 3 repeats of 13 base pair. 4 repeats of 9 base pair and second one is 3 repeats of 13 base pair sequence. So let's see the initiation steps. So here, first here it is 3 repeats of 13 base pair and here it is 4 repeats of 9 base pair. This is Ori C, that is origin of replication. So first in the initiation, first step is DNA A protein. DNA A protein that binds to the 9 base pair, that is 4 repeats of 9 base pair. This DNA A protein binds to this region and it facilitates the strand separation of DNA duplex. That means it unwinds the 13 base pair. It will bind here and it unwinds the 13 base pair and it will be it will lead to the formation of a bubble. Results of the formation, unwinding of 13 base pair, and it will lead to the formation of a replication bubble. So here it is 5 prime, 3 prime, 3 prime, 5 prime direction. Then DNA C is added. DNA C protein is added, and then DNA C and binds to DNA B protein. DNA B means helicase. DNA B, it is helicase. So, in the replication bubble, 5 prime, 3 prime, 3 prime, 5 prime. The this helicase enzyme, it will bind to both strands. This DNA B helicase, it's DNA B or helicase, its main function is it break down the double helix. It break down the double helix and it can break down the hydrogen bonds between the bases. At the same time, at one time, 2 helicases are added and bind to 2 strands. So, one will move in left, this will move in left and this in right direction. So, in the 5 prime 3 prime direction, and separate the 2 strands in both directions. That means replication is bidirectional. So, it will move in 2 direction. This is in 5 prime 3 prime direction, and it will be in 5 prime 3 prime direction. So, that means the replication is bidirectional. So, DNA C binds to DNA B protein and it results in the opening of DNA B ring. Then DNA C is released. After that, this DNA C is released. Then this DNA B bound to the single stranded DNA. Then this helicase, it travels along the single stranded DNA in the 5 prime 3 prime direction. It travels along the single stranded DNA in opposite direction in the 5 prime 3 prime direction. And it causes unwinding and creates the replication fork. Replication fork means it will create replication fork. This is replication fork. So, there will be the formation of 2 replication fork, 1 and 2 replication fork. Then after that, after the formation of replication fork, after the formation of this replication fork, we can draw like this. This then SSB protein, single stranded binding protein, will bind to the single stranded DNA and it stabilizes the strand separation, or it prevents the strands from renaturation. So, single stranded binding protein, it will bind. So, here it will be helicase, here it is helicase, and the single stranded binding proteins will bind. These are single stranded binding proteins, SSB proteins. So, these proteins will bind to the single stranded DNA and stabilize the strand separation, or they prevent the strands from renaturation. So, this unbinding of DNA, it will cause torsional strain. Torsional strain in the DNA, and this is removed by topoisomerase 2 or DNA gyrase. So, after the binding of SSB protein, there will be unwinding, and this unwinding of DNA causes torsional strain, and this is removed by topoisomerase 2 or DNA gyrase. So, this topoisomerase will bind here. Here topoisomerase will bind. Here it is helicase, then the single stranded binding proteins, and topoisomerase will relieve the torsional strain. So, that is initiation. Next step is elongation. It is elongation. So, in the replication fork, in elongation a primer is needed. primer is a short segment of RNA. It is a short segment of RNA. Here the enzyme primase. Primase is also known as DNAG. DNAG. This primase synthesizes a short stretch of ribonucleotide, which is about 10 to 60 nucleotides. 10 to 60 nucleotides. So, at the point of elongation, a primer is needed. Primer is a short segment of RNA. This primase enzyme synthesizes a short stretch of ribonucleotide, about 10 to 60 nucleotides. So, this is the parent strand. Here 5 prime, 3 prime, 3 prime, 5 prime direction. Here the helicase will come. It is helicase. Then there will be addition of primer. This is primer, and here it will be the new strand will be synthesized in the 5 prime 3 prime direction. That means DNA polymerase can add nucleotides in 5 prime 3 prime direction, and it will be here like this. Along with this primer, this DNA polymerase extends this primer. So, after the primer is added, this DNA polymerase can add nucleotides in 5 prime 3 prime direction. So, DNA polymerase can add only in 5 prime 3 prime direction. So, this here it will be like primer, this strand, then again primer, strand, again primer is added, and this strand is synthesized in this manner. So, here it will be in fragments, and these fragments are known as Okazaki fragments. This is leading strand and this is lagging strand. So, a primer is added by the enzyme primase. Then after this, this primer is extended by DNA polymerase 3. DNA polymerase 3 synthesizes the new DNA strand in 5 prime 3 prime direction. Then there will be formation of leading strand and lagging strand. Leading strand is synthesized in 5 prime 3 prime direction. This is in 5 prime 3 prime direction. Then along the replication fork. It is along the replication fork. Then lagging strand synthesis occurs in discontinuously in short segments. These segments or fragments are known as Okazaki fragments. These are Okazaki fragments. This lagging strand means it moves away from the helicase or it moves away from the replication fork. It is formed discontinuously. Leading strand means it follows the helicase or it is towards the replication fork. But the lagging strand is away from the replication fork and it moves away from the helicase. It is formed discontinuously and the fragments are known as Okazaki fragments. So that is about elongation. Then this after this RNA primer is removed. After this RNA primers are removed. This is removed by DNA polymerase 1. DNA polymerase 1 removes this RNA primer. That is the main function of DNA polymerase 1. It removes the RNA primer. Then if the RNA primer is removed, there will be nick or gap. This gap or nick is sealed by DNA ligase. And there will be the formation of phosphodiester bond between 3' OH group of one strand and 5' phosphate group of another strand. So, DNA polymerase 1 removes the RNA primer and the nick formed by this is sealed by DNA ligase. And there will be formation of phosphodiester bond between 3' OH group of one strand and 5' phosphate group of another strand. So, that is elongation. Next step is termination. Termination. Termination means the two replication forks, these are moved bidirectionally in opposite direction. Then they will meet at a position. They will meet here. And this point is known as terminus region. Terminus region. So, the two replication forks move directionally, bidirectionally. They are going to meet at a particular point, and this point is known as terminus region. So, DNA replication terminates at the terminus region, and it contains multiple copies of 20 base pair sequence. It is called terminus or ter sequence. This ter sequence serves as the binding site for TUS protein. TUS means terminus utilization substance, and its top helicase, and simultaneously DNA replication is terminated. That is termination. So, this is about DNA replication in prokaryotes. This is today's topic. Thank you for watching.